Hello, this is the founder of ideaXme, and I'm here with somebody who moves the human story forward. Who are you?
I'm Dr. Ellen Jorgensen. I'm a molecular biologist, and I'm aiming to democratize bioscience.
What is your objective for doing that?
Well, the objective is to get as many people possible engaged hands-on in what I would call modern biotechnology or bioscience. So all of the new technological advancements, mostly based on DNA science, that are moving us forward in the world in terms of all sorts of biomedical technology, biomaterial technology, agricultural technology, you name it.
Can you take us through the key advances within this area?
Well, I think some of the recent ones that have made the news are genome editing systems, where they act almost like word processors for DNA, although I kind of hate that analogy because it's very simplistic, but it does kind of give you a visceral feel of what the difference is between this technology and the technology we used to use, which was much cruder. There's also a movement to be more high throughput with these sorts of experiments with all of the different technologies that are coming together. We have AI, we have imaging technologies, we have the ability to see atoms now. It's really amazing how fast things are moving and how they're playing off each other. So, for example, in medicine, we're now reprogramming people's immune system cells to attack their own cancers, which was something that was unthinkable even a decade ago. So that's the sort of thing that I'm interested in, taking materials that are produced in one species and producing them in a very user-friendly and cruelty-free way. So the whole movement for in vitro meat production. I mean, there's just a lot of wild ideas out there that are finally coming to fruition. It's a very exciting time to be a scientist.
Can you talk to us a little bit about the theories behind CRISPR technology and how it is done?
Well, I like to think of it as kind of like a guided missile. It's a two-part system made out of proteins, which a lot of biological technology relies on enzymes that do work, which are basically proteins. And in this case, it's a combination of protein and a nucleic acid called RNA, which is a very close chemical cousin to DNA. And the beauty of it is that the protein never changes. It's pretty much the same, whatever species you put it into, whatever editing you want to do, it doesn't change. And that's kind of the expensive, hard to change part of these systems. There were some previous systems that did genome editing, but they were clunkier because you had to change the whole thing. In this case, it seems to work in every species we've tried it on. And the targeting mechanism is this little piece of RNA that's you can buy for $100 and get in the mail within a few days. So it's very easy to do a design, build, test cycle if you're trying to test a product. Say you're trying to test a new therapy where you're trying to edit away somebody's like a bad gene of some sort. You can do your experiments, original experiments, without too much outlay of time and materials. It really kind of speeds up the whole process. The way it works is the targeting mechanism targets this protein to a specific place in your DNA, and it makes a cut through both strands of the DNA. And that's a catastrophic event for a cell. So it sends in its natural repair processes, and you can kind of hijack those to get them to do what you want. So it's a combination of something external and something internal. And So far, it's been used mostly in cells that you can take out of the body and put back, like the cells I was talking about, the immune system cells that you reprogram to fight the cancer. CRISPR is being used to reprogram them. So that's an example of its biomedical use. It's also being used in agricultural tech to more efficiently produce genetically modified plants and animals that have valuable characteristics like drought resistance. It's pretty much a tool that any workflow that involves any kind of messing with DNA, CRISPR is important for right now, which is why it's such a big deal.
Where do you see this going in the next 10 years? Best case and worst case scenarios?
Well, I mean, the real hot button use of it, of course, is in human engineering. So that's being discussed over and over again, both within the scientific community and in the press, the whole designer babies thing. And right now, the bottleneck for most sort of therapy with CRISPR, so say a person has a disease like cystic fibrosis, which we know what the cause is, we know exactly what the letter in the DNA we need to change. And wouldn't it be great if we could go into that person's lungs and change it over to a more normal lung? But the delivery is the problem, because you have to get it inside the cells. And so that's been the hurdle for gene therapy and a lot of other advances, is how do you target the right cells? How do you get the system inside the cells? And right now, we're using technology that we've used for many years in the past. So you can deliver it with a virus. You can deliver it as a nanoparticle. But nobody really knows how efficient those deliveries are. There's also a problem with potential off-target effects, where it edits where it's not supposed to edit in a small proportion of cases. But if the edit is something catastrophic, like if it produces a cancer cell, then that's bad. So they're working on those problems. However, the easier way to go about getting rid of genetic diseases is to do it at the embryo level. And that's the level that is very controversial. There are people who don't believe in embryo editing for a variety of different reasons. They don't think that it's a procedure that is morally responsible. It's also something that would have to be proven extremely safe in order for it to be used wisely. But I think the biggest fear that people have is that it is going to work, and it is going to work very well. And we've had some preliminary experiments that seem to indicate that it does. Where do we draw the line?
Yeah.
So right now, everyone is talking about editing away diseases. But if you kind of take that further and you say, well, what is considered something that needs to be edited away? Or what do we want to think about enhancements? If you think about the lengths that people go through to make sure that their kid gets every advantage in life, would you want to make your kid smarter if you could, or taller, or more athletic, whatever? And also just the idea of changing that bell curve that is the population, where you have people on the two ends of the bell curve that may be very unusual in one way or another. And is it good for society in general to make everybody more uniform? And this argument can be applied to a lot of different things, not just genome editing, but it is a very real thing. And then the last thing is the economics of it, because so far, the only way you can do this is through in vitro fertilization, and that's like a $30,000 procedure. So are we going to create a society that has different classes that, based on your economic, your wealth, are you going to be able to ensure that not only you, but your descendants forevermore will not get a genetic disease, while people that can't afford that procedure are doomed to have their families riddled with that disease forevermore. So who pays for it becomes a little bit of a hot button as well.
A number of reports have come out, including the report from the National Academy of Sciences and the National Academy of Medicine. But there's one thing on genome editing and the things that we need to be looking at, and recommendations and so on, touching on the things that you have mentioned. But there's one thing having a report. How can this thing, this process, be overseen? What are the practical tools that we can put in place to make sure things are being done properly?
Well, it's a very interesting question because in certain cases, the edits, the other, which I didn't mention, the other controversial thing is editing entire species. There's a variety of CRISPR, sort of a different flavor of it, called a gene drive, which is sort of like a perpetual motion machine for genome editing. And if you have one individual organism that has this, it can spread a genetic characteristic through the population extremely quickly. So it overrides normal Mendelian inheritance, and all of its progeny, no matter what organism it mates with, whether they have the gene or not, all of the progeny will carry the new edit, and all of them will give that. So within 10 generations, you can completely change over a species or even eliminate it if you do some sort of lethal edit or an edit that makes the organism less able to reproduce. And this is actually being discussed for the mosquito strain that carries malaria and Zika. So in that case, it becomes a matter of how do you bound something like that? Like a mosquito isn't going to recognize the boundaries of a country or a city. And you can say, well, we want to deploy it somewhere in this particular region, but there's no way once you deploy it that the problem doesn't become something that is potentially worldwide. And so the United Nations has issued some directives on that, but they don't necessarily have any enforcement power.
Yes.
So, the question becomes who regulates this stuff. Yes. And who should be able to regulate it. And that's a huge question. I don't have a ready answer for that, but it's something that's up in the air right now. I mean, what I will say, and I think what you're getting at, is that if you want the public to weigh in, they have to know that it exists. And if you ask the average person on the street, unless they're like glued to the Discovery Channel or something, they are not necessarily going to know that all these things that we've talked about in science fiction for 100 years are now really close to happening. And that's one of my missions in life, is to kind of spread the word on how fast the science is moving and tell people they should start thinking about what their opinion is on it.
Can you tell us about your work in this area?
So right now, I'm on my second community lab. It's called Biotech Without Borders. And one of the nice things about these laboratories, which are essentially, I define them as a lab that's standalone. It doesn't, it's not part of a university or a company. It's a nonprofit entity that exists to serve the public. So one of the things we do is we provide lab space for people that want to do projects that may not have access to a lab. But the other important part of the mission is outreach in the form of hosting all sorts of lectures and public forums around the technology and getting people involved in it hands-on with classes and workshops. And I think that's critical because a lot of times I feel if it's a technology I don't understand, that this technology is being imposed upon me. And it's frightening because you don't really understand how it works. You see it cropping up in all the daily things that you do and products that you use in medical treatments, everything else. And it's kind of scary. And if you actually do some of it, even at a low level, hands-on, I think it has the potential to demystify it. So that's one of the things that we do at Biotech Without Borders is we invite the general public to come in and actually do some of this stuff hands-on. So I have a CRISPR workshop where they actually knock out a gene in the yeast genome, and the yeast turned pink because this red intermediate in a metabolic pathway builds up. And we also have hosted a number of public forums around CRISPR. There's an excellent public forum that was prepared actually in response to the report you were talking about by the Museum of Science in Boston. And I've been part of the prototyping of that forum. And it's all around getting people to look at a few scenarios that, may or may not be likely, and just asking them to think about how they feel about it. And then everybody sort of shares out at the end. And I think that's a really useful exercise. And the scenarios are things like, well, if you had a genetic disorder, would you be open to some sort of CRISPR-based therapy? If you had a child, would you be open to enhancing them? If you could make them more athletic, would you do that? And then there's one around if people have a genetic disorder that is a burden on the healthcare industry. I think they use sickle cell anemia as the example because people have a lot of hospital visits with that. Do you think that they should be given a choice of having gene therapy if it gets to the point where it works with CRISPR, gene editing therapy, I guess? And who should pay for it? Should it be mandatory? All that kind of thing. There's a lot of sticky questions.
You gave a talk in Dubai, I think it was this year, when you spoke about a development that you thought might happen within China in the next 2 years.
Well, it's interesting. The Chinese have really embraced biotechnology. And I really get the sense that they really want to be the world leader. And the government has put a lot of resources behind it. They seem a lot more fearless than we are about going forward with stuff.
What's your opinion of that?
I think they're good points and bad points. I like the fact that they're moving the needle forward. They often don't do as good a technological job as we do. So, for example, the first 2 editing experiments with human embryos were in China, but they were non-viable embryos. We had to wait for the one in the United States to be the first, which was almost a year ago now. And that one was supposedly an implantable quality embryo, although it hasn't been repeated as far as I know. And the Chinese have done experiments in monkeys now, and so we're waiting to see how the monkeys develop. I guess the prediction I made was that I thought that the first CRISPR'd baby would be born in China, and I still think that because there's a big push to use these technologies to better the health of their population, and they have the kind of monolithic government structure that's able to make a decree of, make it so, and it becomes so without a lot of debate. So,
you in one of your other talks, you mentioned legal battles around CRISPR.
Well, there was something just recently that there was a resolution in the legal battle. I didn't read the news report, so just a friend of mine told me yesterday that there had been a settlement in the case in the United States between the Broad Institute and Berkeley.
Yes, yeah.
And I think they came down on the side of the Broad Institute again. But I don't know. I mean, you have to check that. You have to fact check that because, again, I meant to read it and I didn't get to it before I did this interview.
Do you foresee?
But it's been a long ongoing battle. And normally, when two universities have sort of overlapping technology patents, the lawyers will get together and they'll divide up the pie. I think it's indicative of how transformational CRISPR technology is and how valuable it is that they went through such lengths to go head to head for so long. And it's costing them a ridiculous amount of money in legal fees. But what's interesting is that all sides are making it free to use for people who are doing research.
Oh.
So it's not like the technology is being suppressed or closely held. Everyone's using it.
So like open access?
Well, not so much open access. It's more, it's readily available to anyone who is doing research. But if they invent something that's patentable, that's when sort of the licensing fees are going to come into play.
As I've explained to you, the ideaXme audience is from subject matter experts within the specific fields in which we interview people, but also the general public. Are you able to recommend some papers for people within your area of interest, and also for the general public, maybe a book or two to come up to speed?
Well, Jennifer Doudna just published a book. Again, I'm sorry, I can't remember the name of it, but it was on the bestseller list, and it's about basically CRISPR and genome editing technologies. She also, for people in the field, just came out with a review quite recently on the subject. And of course, she's the subject matter. She and Feng Zhang and a number of other scientists who were credited with inventing or discovering the technology are the subject matter experts that I would read stuff from. I think that National Academy's report might be actually quite interesting to read.
Yes,
I would recommend that as well. And that's generally written at a level that the general public can understand. And those are usually quite thoughtfully written. So there's also some very interesting stuff online. The New York Genome Center has a series of videos, and they published one on CRISPR. And the idea is that they have someone explain it at the level of different people. So it starts out like, I guess, the level of a 10-year-old, and then the level of like a high school student, then the level of a college student, and then maybe a graduate student in the area, and then an expert. So it's like a series of like 5 different explanations of the same technology, increasing in complexity and scope each time.
That sounds brilliant.
Yeah, I thought that was a really nice way of presenting the material.
So what are your plans with Biotech Without Borders over the next 5 years?
Oh, man, I would really love to expand our reach. One of the things that really struck me, I've been attending these sustainable development conferences at the UN, and there are so many countries, particularly in sub-Saharan Africa, that really could benefit from having more capacity for biotech internally. And there are a number of really amazing people working to make that happen. And I would like my organization to be able to participate in that in some way. And I'm trying to work towards that. But it's an area that I don't know that much about in terms of delivering nonprofit services or liaising with people. So that's kind of the area that I'm looking at, is to become a little more global than I was with my first nonprofit, and also to I've gotten really into teaching the teachers. So we have a great program called Hack the Helix that we're expanding this year that allows New York City public school teachers to come in and have access to a real lab and just swap tricks and techniques with each other and maybe broaden what they're teaching, because this generation of students that's in high school now, the science that they're going to come out into, if that's where they're interested, is so much more sophisticated. I mean, when I was in high school, I was making macaroni models of DNA, and these people are doing experiments that, 20, 30 years ago were Nobel Prize winning experiments, and it's part of the high school curriculum. So it's changed a lot. And in order to prepare them for this, to be the scientists of the future, I think a lot of the more savvy STEM teachers are really trying to open themselves up more to hacking the helix.
Do you think that any aspect of what you do could be brought online to online classrooms, or do you think it's necessary, just because of the nature of some of the dangers, I guess, for the teacher to be present in the classroom?
This is a very interesting topic to me, and actually I've been thinking a lot about it in the past couple of days. Because I very often get requests, particularly from people in other countries who can't come to the United States and take the classes at Biotech Without Borders. Is anything available online?
Yeah.
And after a great deal of internal soul searching, I've decided I think I am going to put some classes online.
Oh, fantastic.
And the reason is the information is available sort of piecemeal anyway.
Yeah.
So it's not like I'm revealing any kind of a secret. And if it's within the framework of what I'm doing, it always comes with a discussion around, ethical behavior and safety and everything else. So I would rather come at people with everything bundled together like that, rather than have them just sort of pick and choose techniques and not think about things like biosafety or biosecurity. You can't really contain this stuff. I briefed a UN disarmament committee once, and I just remember their faces when I said that you could put DNA, you could spot it onto a piece of paper and mail it anywhere in the world, and their mouths kind of dropped open.
Yes.
It's not something that you can suppress. I think that a lot of the techniques that are coming online now make it easier to do. The thing that's the hardest is actually figuring out what changes to make in an organism to get whatever result that you want. So the effectiveness of what someone does, everyone is always thinking, someone is going to create this super pathogen. It's not easy to create a super pathogen. If you think of all the millions of microbe species there are, and how Mother Nature is experimenting with them all the time, you only have a handful of what the FBI calls select agents, things that are so dangerous that they have to be tightly regulated. So if Mother Nature, with all her power, has really failed to create huge, huge threats in a big way, it's not that easy to imagine an amateur stumbling onto something, or even deliberately creating something. And so it's there. With the H5N1, when that flu virus, the papers were published that said what were the specific letters that were changed in the flu virus to make it super infectious, and there was this big debate over whether or not that should be published. In the end, they let it be published because the information was extremely important, and nobody could figure out who would be the gatekeeper for it.
Yes.
So, obviously, health services all over the world need to know if they're monitoring flu strains in their country.
Yes.
And 3 out of the 5 mutations start appearing. They know they're 2 mutations away from a potential pandemic. You can't suppress that information. Uh, it's always rough. If a technology is sufficiently powerful, it's always going to be dual use. And you sort of have to take a deep breath, do the best you can. So I applaud DNA synthesis companies not just sending out any sequence that anyone asks for. I applaud that there's a certain amount of gatekeeping there, that they're not going to send someone, genes for ricin or smallpox without, very good reason. But at the same time, I don't think that it should be heavily regulated.
What have the key opponents been, or who are the key opponents? What are the barriers to this openness that you are promoting?
Well, it's interesting. I think the biggest barrier to open science in general is the reward system. So right now, if you're a young professor at a university, your tenure is dependent on publishing papers that are yours and that are unique and that are important. So if you are working on something and you're discovering something, it's not to your advantage to share that knowledge and have someone with more resources potentially scoop you. So there's a built-in barrier to collaboration in that if you want to get ahead, your university wants you to be the first author on the publication or the last author on the publication, not stuffed somewhere in the middle. And in industry, it's based on patents. So if you have a technology that's patentable, you're likely to get investors because they figure that you have a competitive advantage. If you make it open source, you have to be very clever about how you go about marketing a company. So Arduino comes to mind, and the thing that makes Arduino work is that, yes, it's open source, but it's at a price point and a difficulty point where most people would rather just buy an Arduino rather than trying to make it from scratch themselves. And it also, they have run into competition with Raspberry Pi and a whole bunch of other small computer chips. So the whole system is set up so that openness and sharing is not encouraged. That really is the biggest barrier to this sort of stuff.
If you were given the job of heading up a worldwide committee, but unlike the UN, you were able to enter into implementation of the findings of a report, let's say, what would you do as head of that committee in ensuring that the growth of exponential technologies can be managed to the advantage of everybody and to reduce the risks?
Well, there's an interesting experiment being done by a professor named Esfelt at Harvard. He's working with gene drives. And his whole thing is he's trying to create a framework for the work to go forward transparently. So the communities that he's working with, there are a lot of town meetings, everything's documented, all the documentation is made public. So there's a level of transparency that you normally don't see in scientific research associated with his project. And I think that's a step in the right direction. I believe in democratic societies, but I also worry that that the public can be swayed by fear.
Yes.
And that there are a lot of people that are really good at fear mongering. It really makes one of the reasons I got into this field was I was very frightened by how effective fear-mongering has been in GMOs in food. There's absolutely no evidence that food that contains modified DNA is bad for people to consume. The idea that somehow the edited DNA is going to get into your DNA is so outside the bounds of what we know about about how, food enters the body and everything else, hundreds of years of research, that it's laughable. Yet, I see this same publication with rats and cancer cited over and over again. And when you actually pull the paper and you read it, it really says no such thing. The data is completely inconclusive. They use, possible might, all these really soft words in the discussion section to describe the findings. They're not statistically significant. And yet this one paper gets quoted over and over again. And no one ever bothers to click through and actually read the paper, or has the scientific background to understand the terminology and the way people are talking in it. And that drives me nuts. And so I don't want the torches and the pitchforks to come up the hill and basically shut down this research, which is really critical, I think, to our survival on the planet. And yeah, I know a lot of people don't like the idea that we're going to use one technology to fix the mistakes of another technology, but we've either got to reduce the number of people living on this planet drastically, or we're going to have to find high-tech solutions to feeding everyone and keeping everybody clothed and housed and comfortable, because there just isn't enough going on right now to do that. And, this imbalance of technology is also frightening, where you have countries that have it and countries that don't. And if it gets sufficiently imbalanced, that's a recipe for political unbalance too. So I'm a little worried about that. So I would like to see a more equitable distribution of the technology. I would like to see, and there's a lot of work being done in certain countries to try and educate everyone that, GMO food doesn't make people sick, because literally whole economic systems are in danger of going down if there's a crop that people are depending on. One good example is the papaya crop in Hawaii, which almost was obliterated by a particular disease. And the fix was in a genetically modified papaya, which is perfectly safe to eat, doesn't do anything bad to the environment. It's just resistant to this virus. And yet the anti-GMO lobby almost shut it down. So that really frightens me. And I want to do something about that. It just kind of not only just spreading the word about the science, but getting people to actually do some simple genetic engineering and realize it isn't this incredibly evil brew of witchcraft. It's just another technology. A rock is dual use. You can build something with it, or you can hit someone on the head with it. So people should not be scared of this stuff, and that's one of the big reasons that I like these community labs.
What is it in your history that brought you to this point? What drives you to do what you do? You did a PhD in molecular biology, but when did this passion, this fascination begin?
Well, I had a very typical career for most of my life. I mean, I worked in the biotech industry for many, many years, everything from inhaled insulin formulations to looking at tobacco-related disease markers and virology. It just after a while, you get to an age where you just don't want to be a brick in the wall anymore. And it became obvious that actually the research I was doing, I wasn't like on the road to a Nobel Prize or anything. I mean, I was doing solid work, but it wasn't, terribly exciting in the sense that, I was focusing on a particular disease state or whatever. But when I heard about this movement to democratize biotech, also what I was just saying came into play, in that I felt that there was, at least in the United States, a profound anti-technology, anti-intellectual movement, and it scared me. And so I got involved in what we've called the DIY bio movement in the United States. And because I had started laboratories in the industry, I had the knowledge and the skills to start a lab fairly quickly. And so even though there were other people that I would say were more important to the movement back then, the group in New York, we managed to open a lab before anyone else, just merely because I had the skills. And I got involved very much by accident. I was reading one of these News of the Weird columns in a local paper, and I read something about people trying to make glowing green yogurt and having labs in their closets. And I thought, well, gee, this is something that I do for a living. It's great that people are enamored enough of it to want to do it as a hobby, and maybe this should be supported to kind of counteract that anti-tech feeling. And that's what drew me in originally. And then two of the people that showed up at our original meeting were students that couldn't find any lab space to test out some ideas that they had. And I saw myself, because I had to go kind of from lab to lab, seeing if one would take me on as a volunteer when I was younger. And I thought there should be a space for them to test their inventions. And, over the years, we've learned that these labs are good for a number of things, including very early proof of concept experiments, where you get someone who's an entrepreneur who may not have the most typical career path, or maybe it's even someone who's not a scientist but has partnered with a friend who is, so that they can actualize their ideas. And it's nice to have people thinking outside of the box or doing things on the art-science interface in these labs, because I think it really jumpstarts creativity.
You've been featured in many of the world's media, and you talk regularly on this subject, advise the UN, so you meet an enormous amount of fascinating people. Who would you like to meet, and what question would you like to ask that person? Obviously somebody maybe you haven't met before.
Well, this is going to sound really strange, but my first passion a long, long time ago when I first went to school was astronomy. And I would really like to meet someone from another planet. I would really like to know if there's any intelligent life out there. This is something I think about fairly often. Unfortunately, I think what the rule of the universe is that by the time any species gets sufficiently technologically advanced, they end up destroying themselves. And so we're never going to see anyone from another planet that has life forms. But I really would like to know. I would like to meet someone or some entity, and basically ask them what their life is like, kind of how their consciousness is structured, whether or not they have the same sort of emotional circuits that we do. Do they love? Do they hate? Do they aspire to things? That would really be, for me, a really interesting thing to do.
Ellen Jorgensen, founder of Biotech Without Borders, thank you very much for your time. It's been an absolute pleasure.
It's been a pleasure for me too. Thank you.